the work we are doing in Green Spain, in Granada, at Fundación Medina. And especially today, I would like to focus on what we are doing in exploiting the bacterial diversity of our microbial collections and the different approaches we are performing just to expand this diversity. I don't need to stay too much on this first slide because this has already been covered by Joachim in the introductions, but I would like to emphasize, I mean, the special role of natural products in the last decades in being able to provide just the scaffolds or the molecules that have been used to develop novel drugs. Still today, they represent a very unique space compared to synthetic libraries. Not only they have a huge potency and selectivity against targets and just generated by the extended evolution selection, there's still a lot of underexplored microbial sources to be used for the discovery of novel compounds. And of course, for chemists, there are more and more just outstanding starting points to discover novel scaffolds that can be used as inspiration for the design of novel compounds. In fact, recently, just by the last review by Newman and Craig, they have been highlighting that more than 53% of the chemical entities in the last 20 years are derived or inspiring in natural products. So in this context, I mean, today, that's the situation. I mean, the large pharma has been investing for many decades in getting novel sources of drugs from these compounds. Today, there is a reduced interest in the big pharma, and all the activities concentrated mostly in the academic sector and in the biotech programs. This abandoning of pharma just in the field of natural products has seen that there's a lot of expertise that has been lost, a lot of collections that have been disseminated and not been kept. And in fact, today we need to continue to invest in this area if we want to keep the expertise and also keep the sources that have been used for the discovery of novel compounds. And it's within this environment that was established, the Fundación Medina, this non-for-profit private research organization that was established from the origins of the former Merck site that was working in Spain for more than 50 years, working on drug discovery from microbial natural products. And in 2008, when the big pharma decided to continue to work on natural products instead of dismantling all the center, the subsidiary in Spain managed to get all the assets on the former center at Merck had in Spain to be donated to the new institution and to establish a non-for-profit public-private partnership with the government of Andalusia and the University of Granada. So that's why now the center that was in Madrid was moved to the south of Spain. And this was established with a mission to continue to discover novel bioactive compounds and also discover novel therapies that could be used to tackle unmet medical needs. So in this context, the aims of the foundation, of course, are using the assets that we have in the institute, focusing on drug discovery platforms, and we are focusing on four different therapeutic areas. We are working in infectious diseases, in parasitic diseases, especially tropical infections, oncology and immune and neurodegeneration. And today I would like to focus mostly on the type of activities we are developing in managing to use the sources we have and the approaches we have been exploring to discover novel antibiotics and anti-infective compounds from bacteria in the collections. In our case, all these activities being performed in close collaboration with a lot of institutes, with a lot of small companies. Also, the big pharmas are interested in exploring these collections. And as you can see, there's a big network of collaborations going on, not only in the exploitation of novel compounds to tackle gram-negative pathogens infections, but as well in tuberculosis. We're also working in antifungal treatments, and also, as I was explaining, in how to deal with novel compounds to address tropical diseases. And these partnerships are being just sustained by different type of programs, also funded by different consortia funded by European Commission. This has been developed using the natural product discovery platform that we have been transferring to the center. As you can see, this is a multidisciplinary research platform where we have biologists, chemists, microbiologists working all together in all the different stages that are at the end just ensuring that we can manage to isolate and characterize novel compounds just being produced from microbial sources. So this involves the expertise in the microbiology field, in the natural product chemistry, at the same time the high-throughput screening platforms we develop in the center. So with all this together, I mean, the major point that this was addressed before by the other speakers is how we address to exploit this microbial diversity and to ensure that we get just diversity from the perspective of the chemistry, of the structure chemistry of the compounds. So, of course, there's also just how we are going to address the sources we are going to be sampling, the environmental conditions we are using. In fact, there are strong selecting factors that are just defining which are the specific microbial assemblages that we want to exploit. Of course, there are very broad the biodiversity geographic patterns. We also are very much interested in exploring the different terrestrial niches, the plant-host associations, how we exploit the marine environments. And of course, we cannot forget that, in fact, even the majority of the strains in Vermont have not been cultivated. There is also another way to address how we can be domesticating some of these microbial groups. And a final point is, of course, the microbial collections we have been using for many years. We have only been just testing a very small amount of compounds produced by these microorganisms. So there is a way to exploit from a different perspective the microbial collections that are existing in the different centers. So from our perspective, we are to the microbial collections now at the Fundación Medina is harboring more than 116,000 strains divided between fungi, actinomycetes, other bacterial lineages. And we are dealing on these three main different criteria. In fact, we are, for collecting, just relying on the geography, marine and terrestrial sources that we are using. The ecology, we need to understand where we want to look for some specific groups of taxa, plant, root, sediment, water, rocks. So we need to select first what we want to isolate. And of course, taxonomy at the end is key in helping us to decide if this new isolate, in fact, is new for our collection, and we are just contributing to increment this diversity. So as a summary of what we have harboring in the collection, you see there just a map summarizing the different sources, geographical sources we have been working in the last 20 years. And as a summary, more than 32% of our collection of fungal strains are concentrated on plant endophyte. We have been sampling all over the different places, specifically working with botanists, guiding us in going into endemic plants, just to try to isolate the very unique novel strains. We have been working extensively, of course, in soil and plant rhizosphere actinomycetes. And of course, in the last 10 years, we have been also exploiting the marine environment, not only from sediments, but at the same time from invertebrates. So this is the source that we are using to exploit in our collections. This is just a snapshot of the kind of diversity of representative of the subset of less than 4,000 bacterial strains, where you can see there's a broad diversity of actinomycetes and other bacterial lineages represented in the collection. And in fact, there is a long history of success of novel compounds that have been produced in the last decades by these strains. Of course, there are strains compounds that have been for many years in the clinic, in the market. From this collection was the first carbapenem that was discovered, the antifungal caspofungin. Let me see. Whoops, I managed. That's the pointer. Caspofungin. Even the first statin was discovered in our center. And besides many other antifungals and immunomodulators, even insulated mimetics, I would like to emphasize in the last decade the discovery of the first platensimycin, the completely novel scaffold, new fatty acid inhibitor, and more recently a new topo-2 isomerase inhibitor that was produced, very unique strain in our collection. This is a complex structure that in fact was produced by a novel strain of Kipdelosporangium that was discovered as a novel species. And this was identified from the screening of more than 250,000 extracts from the collection. And this was a very unique strain, and when we look at the collection for other representative of this genus, this really outstanding at a very unique species. So this compound is characterized by having a broad spectrum against gram-positive active bacterial activity and as a potent inhibitor of this DNA synthesis. And the compound, as many other natural products, is not being produced as a unique compound, but also as a family of compounds with different modifications that also is helping quite a lot just to define some SAR in terms of the potential optimization of the compound. So as you can see, this strain that was very unique as a single isolate in our collection that outstand as a completely novel species within this minor genus within the Actinomycetes. So what we are doing into the discovery of antimicrobials, of course, we are just playing with the sources of the novel compounds. And at the same time, we are also combining it with the targets, how we are approaching the discovery of novel antibiotics and the combination with phenotypic screening, just to ensure that the compounds that we discover can penetrate and reach their targets. So in this sense, for the exploitation as on microbial resources, as I was explaining, we're combining on one side the access to an environment, and essentially we're also exploiting quite a lot the isolation of novel not cultivated strain, as I will follow to explain later. But at the same time, we are also putting a lot of emphasis in going into our microbial collection and being able to untap silent genes that have been completely overlooked in the past in the way the screen was approached. So that's one of our major interests. So from the perspective of how to address uncultivated microbial populations, so of course you are very much aware of the recent iChip approaches just to try to trap and domesticate species that are in the environment and bring them into the lab conditions. So we have been working on designing some what we call the diffusion sandwich system that was inspired on the specific chambers developed by Kim Lewis a few years ago. And in this case, this array ensures that you can manage to have 384 small rods of gel and gum that is going to be ensuring and trapping just cells, living cells from the soil suspension, having them been exposed to the signals in the environmental source that has been used for the isolation of the soil suspensions for 2-3 months, enabling just the small propagules to develop within these plugs, and then later on being able to have these single individual cells that have been managing to grow within these beds that you can see are not more than 1.6 millimeters in wide. So retrieve these strains into as pure cultures, little by little domesticating them in laboratory conditions. So by using these approaches, of course, we have been managing to recover also strains that could also be isolated from standard procedures, but also we managed to get enrichment in some groups of taxa that cannot be recovered by traditional isolation procedures, but that can be detected, of course, when we go into the pyrosequencing of the whole environmental sample. So we could manage to enrich in some groups of Genamatonematous, Microbia, and Acidobacteria. And I would like to show you just two examples that we have been managing to put into culture, and also the ability to recover as well novel species coming from these approaches. This is one of the cases, so one of new species of Luteolibacter. As you can see, as before there was communication between strains, we managed to get this description in the microelectron microscopy from these strains, and we managed to put them into culture, and in less than 1 month managed to get these strains into pure cultures. Another example that was extremely interesting is we managed to retrieve one of our isolates that just when we matched the genome from these strains, it was just matching with a long set of uncultured strains from the genome Atymonadites. And in fact, this is the first time one species from this group was managed to put into culture. In this case, the strain that has been described as a new genus, and now we have just completing the description of this strain. Initially, it took about 3 months to have this impure culture, and little by little, the strain has been domesticated, and we can manage to get this level of growth in less than 15 days. As you can see, is a gram-negative aerobic and no-motillar road, and is really not tolerating a rich culture media. One of the reasons why we cannot isolate them in most of the isolation media have been used in the past. And when we have been just exploring the genome of this new genus and new species, we have seen that in fact they have a very large chromosome with more than 6 megabases. Is rich in GC content. And despite this chromosome is not completely full closed, we have been identifying a lot of secondary clusters associated to secondary metabolites, not only NRPAs, terpenes, lantipeptides, and other complete clusters that are potentially coming from horizontal transfer from other sources. So at this moment, we are exploring the compounds just to see if we can manage to have some of them being produced in the conditions. Nothing has been optimized at this moment. We are just testing both solid and liquid media. And there are potential novel compounds being detected by mass spectrometry produced by these compounds and in very, very small amounts. That is another challenge in dealing with all these strains. So, as you can see, it's not only just the point of having in combination just the gene pool in terms of having the strains or the whole genomes. We also need to manage how we are going to be producing, because at the end, if we want to get novel chemistry, we need to manage to get this being produced, no matter it's in the original producer or just in the heterologous gene expression system. And today, we need to rely on all these other disciplines that, in fact, is not only just the microbiology, but also anything related with metagenomics to be able to be independent of the culturing of the strains, of course, of the synthetic biology that is going to help us just to manipulate all this cluster and reprogram all this information, and also all the analytics that is helping us to address very, very quickly in the process where we are just dealing with very interesting novel molecules. So in this sense, the approach we have been following with all the collections and all the strains in our collections is to address how we can manage all the culture-based approaches in terms of not only just to increment the number of conditions and the stress conditions we are using to exploit these secondary metabolites. We are playing with the addition of chemical inducers of any types just to see we can just turn on these secondary metabolite pathways. We are playing with epigenetic modifiers in the case of fungi, and we are getting novel molecules being produced with different types, and also how these strains are cross-talking in trying to work with inculturing and not having any more axenic culture into cluster. So these extracts just coming from these different approaches have been enriched our collection of natural product extracts and are normally being used in the different programs for the discovery of novel compounds. So as a starting point from these extracts and fractions, we normally perform a high-throughput screening. In the case of anti-infectives, these are empiric assays on a broad panel of key pathogens. Very early in the process, because it's always the problem of the identification of non-compound. We introduce not only the hit confirmation, but as well the LCMS, the replication against our own internal databases to select only those that are promising molecules and can ensure novelty in the compounds. And then later, in less than 3 to 6 months, we can manage to have just the pure compound and the structure elucidated. So, and confirmation if we have a novel chemistry of interest related with the activity we have. So, just managing the platform that I was explaining to you, combining the collections, the screening platforms, the natural product library, and the chemistry and the expertise in the field. We have been managing to run different programs, just addressing different groups in the libraries. On one side is the microbial collection that is focused on marine environmental samples and marine bacteria that you can see has been originated from sediments, from actinomycetes of different origins. There is over than 50,000 bacteria have been exploited, more than 75% corresponding to actinobacteria. And in this case, we have been managing to identify novel analogs just to previously describe strains. It is something that occurs more frequently. So you can identify novel analogs with novel bioactivity that was not described in the past, as well as in the case of this strain of Microbiopsis that was isolated from Adinarian. Of course, there were some of these compounds that were previously described as produced by other bacteria, but we managed to identify 4 novel compounds with activity produced by this strain, among which there were 2 new pentapeptides that we are exploring for their activity. Another source was as well the sponges. So we have been working, just putting together a collection of actinobacteria coming from sponges from Florida. And in this case, we focus on Micrococcus and Arthrococcus coming from these sponges, just to have a very, very targeted approach. And we explored all these strains in microfermentations using a combination of more than 12 media and testing them against a panel of pathogens and a characterization of the active extracts. And in this case, we managed to identify 3 different isolates from the genus Cucuria, as well as Micrococcus, that were producing novel thiazolate peptides from these actinomycetes, so very potent activity against gram-positives. So this is also another source that was previously neglected as producing potential novel compounds. And from the perspective of the antifungals, we also explore. We were very much interested in improving the clinical use of many of the antifungals now that are being used for treatment, not only just of candidiasis, but as well as pergellosis. And given that in the case of echinocandins, and especially and Cansidas was originated in our center, we also looked for the identification of compounds that could be just synergizing and decreasing the therapeutic dose that could be necessary to be used in the treatment of this antifungal, given that there are some side effects and which are relatively toxic. So in this case, what we did is we performed just the screening just against Aspergillus fumigatus, using and selecting all the hits that with 70% inhibition that could be used to decrease the level of echinocandin that was necessary in the screening. By running this approach, we could identify not only just compounds that could show antibacterial, antifungal activity, but as well some of them that were showing a potentiation effect in the case where we were using sub-inhibitory doses of the echinocandin. And these were those that we were interested in exploring. So what I'm showing today is just two families of compounds. One of them is a non-compound that was previously completely just described only as a potent antiviral, but had never been shown to exhibit this effect and decreasing more than 5 times, potentiating more than 5 times the effect of caspofungin. It was produced by a soil isolate in the Azores Islands from a Chitoceratospora strain. And another example that was also used is humidimycin that was recently shown to have a similar effect. And in this case, the compound that was also effective in both against the activity caspofungin against fumigatus and albicans. In this case, this was produced by a novel class 1 glycopeptide that is umidimycin, which is an analog to other thiamycins, and that shows some variety in the positions that are modified with respect to the other strains. This strain was produced by a Streptomyces humidus, that was isolated in Spain in an arid zone. And when we compare the genome of this strain, we could manage to see that it perfectly aligned with the other cluster that were previously described. And the only differences were just in the structure gene that's responsible for the production of this small peptide. In this case, we could manage to identify which were the substitutions when comparing with the non-structures of thiamycin 2 and 3. These two compounds were only described as antivirals, have never been described as synergizing the effect of this antifungal, and this is something that we are currently exploring and to see what the potential mode of action is for this compound. So, in summary, I would like to show you some examples of what the microbial collections are still under explored sources. There are different ways to complement the diversity that are in existing collections. There is key just to not to forget that just understanding what the microbial taxonomy and ecology, and understanding where we are sampling and how we are managing to get novel strains, novel organisms. We need to rely on the genomics and the metabolomics to really infer which are the real clusters and real compounds of interest. And for sure, in the next years and decades, of course, we will need to rely on all the tools that synthetic biology, complemented with the analytics, is helping us to work on this field. And just to give you a snapshot that, in fact, probably is also helping into that, is in that, in fact, we have been only relying into the pharma exploitation just for the treatment and the generation of compounds for application in human health. But all these microbial collections, all these natural products have a huge applications in many other fields of biotechnology. There's a lot of activity going on in agriculture, in identifying novel biostimulants, in flavor for aromatic compounds, food in colors, and also all the kinds of enzymes that these strains can be producing. So there is a huge prospect in this application of these microbial collections. And I would like just to end just by acknowledging all the Medina team that, in fact, have been involved into all this work, and as well all our partners in the different consortia that we have been working with in the last years, as well as the funding agencies of this program. And I would like to take any questions for the audience. Thank you.
Thank you, Olga, for this very nice, interesting talk. And if you come to Granada, you not only have to visit the Alhambra, you also have to visit
Yes.
this very interesting building where Olga and her group is located. It's looking very futuristic. I think we have some time for questions.
Do we have bacteria in your collection? And do we have success with cultivating bacteria by the children?
No, we have not attempted just to cultivate myxobacteria. This requires a very specific expertise. It's just like a cyanobacteria. These are two groups of bacteria that have not been focusing on. And in fact, as you can see, in the parasequencing, there were all these delta bacteria that were there. And with this approach, we didn't apply later on the right tools just to recover them. So that's an open question. If we can manage to get novel mixers by approaching this, I'm not sure if this has already been optimized to do that. So it would be very interesting to explore this. Yeah.
More questions?
How can you find new secondary metabolites from genome sequencing?
Well, this is just normally what the new secondary metabolites are just inferring from what is already known. I mean, what we normally apply in just the normal tools that are normally now available, which are as anti-smash or any other matching that in fact are just establishing what can potentially be just different genome sequences that can be just associated to non-cluster. There's more and more information in the databases, so you are always doing that by comparison with what is already known. What you cannot do from a genome is just to infer a completely novel pathway where you don't know that the sequences are related to any completely novel structures, and that is not related with the typical PKases, terpenes, or just an RPases. So in this case, is really just very, very open in the sense of how you can identify the presence. So in many of these cases, you need to rely on just getting your mutants and getting to more classical methods, trying to allocate where your compound can be related to any genes that can be responsible for that.
More questions? Yes, please.
Excuse me, I cannot hear you. I mean, well, the time to put that into the clinic, I mean, is more than 15, 16 years. So since we started at Medina, we have managed to start our programs, and now we are just starting to generate the lead compounds that need to be optimized as medicinal chemistry programs. So we have not even entered any clinical assays. So we are in some of them. We are in the preclinical optimization of these compounds. We have not been able to commercialize anything, but in other areas, yes, you can get shorter time to the market. I mean, in the agro sector or any other sectors that you don't rely on so much longer times for development of the compounds.
One short question, we continue. Yes, please.
I think you mentioned it a few times when you look at combinations. So I call them microbiomes. They might bring to expression a whole set of compounds that you never touched on when you look at one.
Sure, that's one of the, not only just the co-culturing, but at the same time in these microchambers, one of the points is not to have individual cells, but at the same time having consortia, being able to embed that and making them being transferred into the culture conditions in the lab. And the challenge is how can you deal with that, just to be able to maintain them into culture. And later on, once you detect a compound that is being produced, how can you reproduce that? That's the biggest challenge.
But I think you still start from the idea that the consortium is haphazard, and it is not. It's organized and it reorganizes it. So
Yeah.
you start from there.
Yeah, that's what we're exploring as well. Yeah, thank you.